This conference will now be recorded.
All right, folks, so good morning. We are going to be continuing on with our study of histology for the lab portion of your class. Recall that last class we had met and we discussed the epithelial tissue. We talked about the fact that there are different types of epithelial tissue, whether it is, let's take a look at an image just as a review. This will be helpful regarding the fact that you'll see here, yeah, this is a good image. Just to show you that a very basic image of the fact that as we're looking at epithelial tissue, know that the nomenclature for it would be as a result of the type of cell, whether it's squamous, cuboidal, columnar, transitional, pseudostratified, right? These are all different types of. You'll see the cell structure a little bit different, okay? But what we're looking at is the types of cells, and we're looking at the layers, whether it's a simple one layer or multiple layers. More than one layer would be considered stratified. We talked about the fact that transitional has multiple layers. Pseudostratified looks like it has multiple layers, but it does not. Okay? And particular, we focused on stratified squamous epithelial tissue, whether it was keratinized or non-keratinized. See here, this would be non-keratinized because there's nuclei and live cells at the apical surface. Recall, we talked about apical surface. We talked about the basal surface of the cell, right? And so we'll say that the apical surface many times will be exposed to. And now, in the case of stratified squamous epithelial tissue keratinized for the integument for your skin, the epithelial tissue there, it's called the epidermis. Now, that would be exposed to the air. But as far as any other areas where we have epithelial tissue that's the apical surface will be exposed to either a lumen, a cavity, right? A cavity like the oral cavity, the nasal cavity, or say the lumen of a blood vessel, a vessel, or an organ, okay? And many times we said that the apical surface, when it's exposed to that, it'll be a white area in comparison to the stained cells and tissues and cells of the tissue that we're studying and focusing on. So epithelial tissue. But now we're going to be looking at connective tissue. So when we're looking at connective tissue, we're going to be focusing on a few slides here, and then we'll move on into cartilage, bone, and blood, finishing out the connective tissue. So we'll start off with areolar connective tissue. And here would be a good representation. And recall also that I told you, you should, when you're doing the Google search, you should put the 400x behind it there at the end, so that this way you're looking at 400 magnification for the different tissues and such, because this is what you'll see in the lab. Now you'll see here with this image, okay, so with this image, you're looking at thick lines there. You're looking at the thick and thin lines. The thin lines represent the elastic fibers. Elastic fibers allow for stretching and then recoil, returning back to their normal state of being. Whereas in the case of, say, the pink, the large pink, those are collagen fibers and they're present for strength. And then you'll see the majority of these cells right here would represent fibroblasts, and they would be cells that will produce fibers that are present within the areolar connective tissue. There can also be cells present that are involved in the immune function, okay, just so that you know that. So this is areolar connective tissue. It's also considered loose connective tissue, and it's very airy, right? You see here that it's very different from, so again, it's very different from this epithelial tissue, covers and lines, tightly packed cells, epithelial tissue, avascular. No blood vessels in this tissue at all. But there's tissues that there's in the supportive tissue and the connective tissue below it. Okay? So supportive tissue would be connective tissue, and that would contain blood vessels. But the actual epithelial tissue itself, avascular. So this is areolar connective tissue. You can see many presentations of this type of tissue. Let's go. All right, now we're going to go to dense regular connective tissue. So dense regular connective tissue is the type of tissue where, as you're looking at it in the lab, you're going to see these fibers. These are collagen fibers. And we're thinking of dense regular, we're thinking of tendons and ligaments. Ligaments connect bone to bone. Tendons connect skeletal muscle to the bone. Okay? Dense regular connective tissue. Okay? Now, you'll see here that they're kind of on top of each other, very next to each other, and wavy patterns to them. Okay? Dense regular connective tissue. This is how I represent it in the lab, and I'll show it to you as far as how I would test. Now, again, I won't be testing you all because it'll be with Dr. Massimo, but this is just what my representation to help you to understand and see dense regular connective tissue. You're going to see that it kind of looks a little similar to skeletal muscle, but it's different. Okay, but we'll take a look at that in a few minutes. So dense regular ligaments, tendons, you'll see how it's kind of wavy in its pattern of those collagen fibers. This would represent dense irregular connective tissue. Now you can see it's got the same type of collagen fibers, except they're all going in different patterns. Here's another one here that you can take a look at. Okay, and this would be the dermis. This would represent the dermis of the skin. Oh. So Christy, what is it that you're wondering? You can speak out. Okay. This way I don't have to stop sharing.
If we were going to have a PowerPoint for the stuff that we're going over to look back at.
Yeah, so you have chapter 5, right? So I've posted for you all. So I'm going to share, I'll show you right now. Here's chapter 5.
Perfect.
Yeah, so what I've posted this for you all. So it's just a matter of that I'm not really going over the PowerPoint. I'm trying to show you how you can look at these images online, and it would be very important for you to do that. Recall, I also mentioned histology-world.com, another website that's very, very helpful. But just doing Google searches and Google images, this is, I think, one of the better ways to just study and look at many images. You're welcome. Yeah, absolutely, no problem.
So for chapters 3 and 4, for the notes that we'll be submitting, are we
Yes.
able to take the photos that are used in the PowerPoint and insert that into our notes? Just because I find it easier to kind of have a reference.
If you would like to do that, that's fine. These really, honestly, because I couldn't have the lecture, I wanted to just have you do an assignment that was going to help you. So by you taking your own notes, I would recommend you do this. Honestly, for every chapter, every PowerPoint that you're doing, you should do this. And so this is really, it's helpful for you. So whatever's going to be helpful for you, that's fine. I would do that. Okay. Definitely. Absolutely. So the dermis would be let me show you here, the supportive tissue. So I'll just show you skin. Let me just show you skin for a moment here. Histology. There we go. Now, as you're looking at this image right here, what you're seeing, folks, is that this is and let's make it bigger. Okay. So as we look at this image, okay. So you see here where it says epidermis and dermis, right? And then the hypodermis, okay? Now, the epidermis is the stratified squamous epithelial tissue keratinized, right? This upper layer, you see how that upper layer of this epidermis looks different? Because it's dead cells. There are no nuclei present on this area. And we're gonna discuss this and talk about this, but that's the stratum corneum. It's the layer of dead cells, okay? That's the apical surface. This is the outer area of this tissue, this stratified tissue, okay? It's stratified squamous, epithelial tissue, keratinized. These cells here, the cytoplasm replaced with keratin, okay? So now you're looking at here the dermis, and depending upon what image you're looking at, you can see that the dermis may be a little bit larger as far as the representation here, but just know that the dermis right here is comprised of, okay, really other than right here, see these little bumps? These little bumps inside of the bumps, see the pink inside of the bumps? You see this area right here, this area right here? That would be areolar connective tissue right there. But the remaining portion of the dermis is comprised of dense irregular connective tissue. Now you say to yourself, well, why not dense regular? Well, dense irregular is important because so just take your arm for a moment and just move the skin on your arm. It's firmly attached, and yet you do have movement in the skin, right? And the reason why that you have that movement and that your skin stays, the epidermis stays closely attached to the dermis is because of the dense irregular fibers of these collagen fibers are in all different directions, and yet they're still providing an anchoring to the epidermis. Okay, so that's that dermis area that's dense irregular connective tissue. Okay? Those collagen fibers are going in different directions, but they're anchoring and holding on tight to the epidermis. And then we have here adipose tissue. Adipose tissue is fat. And so what you're looking at here, and this would be a good representation of what you would see in the lab. Let's see, bigger, here we go. So we have here, these are fat cells, adipocytes of fat tissue, adipose tissue, okay? And so they look like kind of like soap bubbles. So when I'm looking at this kind of a tissue, I'm thinking of soap bubbles, okay? And different from if I were pointing to this type of tissue, I would have my arrow kind of in the middle here, right? And I would be saying, hey, identify this type of tissue, and it's adipose tissue. If I were to be pointing to, say, I'm going to show you. All right, now, this is going to look a little similar. So let me come back to adipose tissue, see what that looks like. Then when I show you an image of, and I showed you this before. So take a look at this image here. Now, does it approximate a little bit like the adipose tissue? It does a little bit, right? It really does. But you'll see here that really when I would be pointing to this tissue, it wouldn't be the inner portion because this is a space within the lungs. This is lung tissue. And this is a space between the lungs and in the lungs. And these are called alveoli. And this is where we have gas exchange taking place between the oxygen that we're taking in and the CO2 that's leaving. And know that really I would be pointing to structures like here because this would be simple squamous epithelial tissue. Simple squamous. So let me show you. Yeah, so here we go. The human body is amazing, Christy. I totally agree with you. And very complex and very difficult to learn all about it, but it's fascinating to learn about it. And there's always new information and different things that are going on as far as research is concerned that you can learn from. Pretty cool stuff. So you'll see here, does this look a little bit like this? It does a little bit, right? Yeah, understood. It does, right? But what I'm showing you is that with this here, you'll see that they don't all kind of come together. Some do, but some don't, right? You'll see here that there's spaces, there's gaps, because these alveoli, right, are little bubbles of tissue. And the tissue here that you see all these little lines, it's made up of simple squamous epithelial tissue. This is simple squamous epithelial tissue, okay? That's what you're looking at there. Here we go and yeah, so when you're looking at this image, you're just seeing that these openings, they're not cells, that's the actual space is a space in the lungs where gas exchange occurs, okay? So again, when we look at this is an actual cell. This is an actual cell. Do you see how they all kind of come together? Even though they're kind of oval, weird kind of shape, it's all together, right? It's not the same as when you're looking at that simple squamous epithelial tissue, okay? Any questions with these slides so far? Questions with these slides so far? Go ahead, go for it, Christy. Are they literally connecting? Well, yeah, I mean, when you think about when you think about the adipose tissue, it's connecting, it's a matter of that it's providing support, it's providing an area of where we can connect. So think in terms of the eye, your eyeball. The eyeball is set in the eye socket, right, in the orbit, and it's cushioned by adipose tissue. So if someone is losing a lot of weight, and say they're anorexic, or they're in a starvation type of a situation, their eyeballs will sink in because there's no longer that fat cushion. Or for your kidneys, that can be the same thing. What is the best? Oh, let me finish my response, and then I'll stop sharing. I want to go back to that question. I couldn't see it. Think of the kidneys. Okay, I'll hit you in just 1 second there with that question. But the kidneys also, they are supportive by a fat cushion. And if a patient loses a lot of weight, that also the kidneys can drop, right? They can drop, and the tube, which would be the ureters, which are connecting the kidneys to the urinary bladder and drain the urine to the urinary bladder, they can drop. The ureter can kink, that tube can kink, and this can be a problem. So fat does allow for this cushioning and this protective mechanism. Let me stop sharing for a moment here. Let's see. Okay, hold on. Let me see. Are they literally clear? What is the best way to learn all the different type of tissue? I seem to be struggling with that a bit. Kara, you would be not the only one that struggles with learning the tissues. This is how I'm sharing this with you right now, is how I'm trying to show you how you can look at all of these tissues and step by step go through the epithelial tissue, how you would look at them and identify them, then the connective tissue, that all in a group, and then you could break down the connective tissue into its different forms. So we're looking at the different types, right? We're going to see cartilage and bone have a few different types of cartilage, a few different types of bone. So you can break the connective tissue down into its subsections, so to speak, and look at them and write notes and copy what you're looking at online, draw it. I know that's goofy, and don't worry about it. It's not art class, but it's a matter of when you do the drawing like that and copy what you see as far as the examples, that will help your body, your brain, your memory to retain that information. Okay, so again, too, I know, and this is why I start off right from the very beginning, just showing you these slides so that you have an understanding and get and understand what's going on as far as identification of the tissues. Okay. Yeah. Yeah, so really, I have to tell you that continual, very good. Yeah, so, writing notes and rewriting your notes and rewriting the information from the PowerPoints, very helpful, as well as drawing, very helpful. Coloring, the anatomy coloring book, very helpful. Flashcards, the ones that you create. So you can actually, folks, so here's what you can do. So you can start sharing again. You can take this image, right? You can print it. You can actually print this image if you have color, the availability for a color printer to print out these images of the different tissues and then put them on a flashcard and review them and go from there. Okay, so very, any way that you can do repetition, right? That's what's going to help you to memorize this information. Okay, so don't get frustrated. It's all good. So yeah, so all good, folks. You just have to realize that you're not the only ones that have gone through this, right? And so others have done it before you, others will do it after you. You'll do it now, and you'll put the time in, and you will do all that it takes in order to focus on learning the histology. Okay, that's for the class. All right. Yeah, the atlases are great. Absolutely, very helpful. Okay, so let's now take a look at cartilage. Now, cartilage, so, and also while I'm doing the lecture, while Dr. Massimo is doing the lecture, right, you need to, I am recording. Yeah, it's okay, it's all good. Yeah. While we're doing the lecture, you need to be taking notes. So you should have a blank piece of paper. Don't be sorry. That's what learning is all about, asking questions. It's all good. you need to make sure that you have a notebook, a blank piece of paper, a copy of a PowerPoint, whatever it is, but you need to be taking notes, right? I'm giving you a lot of information. Dr. Massimo will give you a lot of information, and you need to do all that you can to write down the notes in order to retain this information. Okay, so now we are going to be looking at, so let me just give you this just so that you can write this down. So we have, here we go, very good. Now, I'm going to, okay, here we go. So this slide just here, right? And so this is the great thing about Google Images. You can just go throughout here, and I'm going to point out to you the 3 different types of cartilage, and I'm going to talk to you about where they're located. Okay, and again, too, I'm recording this so you'll be able to go back and listen. Hyaline cartilage, elastic, and fibrocartilage. Okay, 3 different types of cartilage. They're very unique in how they look, but similar, but unique. And they also are in different areas of the body, and you'll need to know that. So you'll need to know, so for this chapter, for histology, for the lab and lecture, you'll need to know what the tissue, how to be able to identify the tissue. So identifying the tissue by looking at an image. You'll need to know the function of the tissue, and you'll need to know the actual, and I'm working on identification right now, and also I'm talking to you about location. So you need to know where they're located, right? So I'm going to bypass hyaline cartilage first, and I'm going to go to elastic cartilage. So elastic cartilage, so write this down. So think of the E's. Think of your external ear, your epiglottis, external ear, your epiglottis, and it's in your notes, but I'm just trying to talk to you through, and I'm going to show you examples of. Your eustachian tube, that's the tube that helps to equalize the pressure in your middle ear. So when you go up to a higher elevation, that your ears pop, eustachian tube, okay? So epiglottis, eustachian tube, am I forgetting anything? Nope, we're good. And then, so that's elastic cartilage, begins with the E. And what you'll see here with elastic cartilage is that you're looking at those dark elastic fibers. When you look at this, this is what you're going to see in the lab. This is the chondrocytes, the cartilage cells. They're in a space, it's called a lacuna. Okay, so lacuna, cartilage cell, and you'll see these black areas. Those are the elastic fibers. Elastic fibers. Let's take a look at one here. This is elastic cartilage. The dark lines there, those fibers, those are the elastic fibers. Okay. Cartilage cells, chondrocytes, present within a space. The space is called a lacuna. Another one, elastic cartilage. Very easy to spot, to be honest. This is an easy one. Okay, this is not a hard one. So that's elastic cartilage. Yeah, it is cool looking, absolutely. Now, as we're looking at then fibrocartilage, okay, so we know we're elastic cartilage, thinking of the E's. Fibrocartilage, we're thinking of the intervertebral discs, the discs between each of the vertebra of your spine, okay? Fibrocartilage. The pubic symphysis. So let's take a look and show you. Let's see here. So it's going to be the intervertebral discs. Here we go. Good one. And you can do this, folks. So see the discs here? Let's make this a larger image. What's going on? I'm sorry, question-wise, or? Oh, nope. Okay. Locations. Okay, so let's take a look at here. All right, so with this image here, this is a helpful image, the intervertebral discs, okay? The spaces between each of the vertebra, fibrocartilage. The meniscus of the knee. So you've heard the medial and lateral meniscus, fibrocartilage. The actual temporomandibular joint, the ability for you to move and open and close your mouth, your jaw, that would be the TMJ, temporomandibular joint, fibrocartilage. The pubic symphysis, see this space right here? So we have these coxal bones, your hip bones, and right here in the middle, that's your pubic region, and that pubic symphysis, that joint right there, fibrocartilage. Okay, let's look at fibrocartilage. Close that out. So this is elastic cartilage. All right, so in the lab, in our lab, fibrocartilage, we have the slides for fibrocartilage. They kind of look like this. It's blue stained, right? And you'll see that there's the space, the lacuna, the cartilage cell that's a chondrocyte, chondrocyte in a space there, the lacuna. And here you're seeing the very strong collagen fibers, okay? And it's really, fibrocartilage is very unique in that it's very strong and it's able to absorb a lot of abuse. Okay? So fibrocartilage, it's present in areas that really need a lot of very durable, very tough cartilage. The meniscus, the discs, the pubic symphysis, the TMJ, all fibrocartilage. Okay. Now we go to the last one would be hyaline cartilage. So hyaline cartilage, this is a very unique cartilage in that it looks much different from elastic and fibro, and it has almost like a clear appearance as far as the matrix is concerned. You'll see here that there are these cartilage cells, these chondrocytes in a space, a lacunae, right? You see them all throughout. And you'll see that this is hyaline cartilage, hyaline cartilage, hyaline cartilage, okay? Very unique in its appearance. Hyaline cartilage is every other place in the body that fibro and elastic cartilage are not present. So the ends of bone, right? The ends of bone would be hyaline cartilage. It's called articulating cartilage. It's the cartilage that prevents your bone from rubbing on bone. Okay. Now the cushion, again, the cushion would be the meniscus in the knee. That's fibrocartilage, different from the hyaline, right? But all the bones of the body have some type of covering on the ends. When a bone is touching another bone, that's articulating cartilage. That would be, that's just the term that we describe it as, but it's hyaline cartilage. So your growth plates, hyaline cartilage. How bone grows in length and such, hyaline cartilage, okay? So I'm gonna hit mute for a moment as my dog is. Sorry, someone's at the door and the little doggy likes to bark very loud. So cartilage is the cushion for bone on bone. That's correct, Kara. Yeah, that's correct. And so I'm going to press. You would think someone's trying to kill the dog. So sorry, it'll stop in just a moment. All right, folks. So very good. So now, again, coming back to hyaline cartilage. So really, all throughout the body, hyaline cartilage, except for those areas fibro and where fibro and elastic cartilage are not located. So your nose, fibrocartilage. I mean, hyaline cartilage. Sorry, hyaline cartilage. Okay. So hyaline cartilage is present throughout. The growth plate again allows for bone to grow in length, okay, and in size. And so really know that. Also, we'll be looking in depth in the chapter regarding the skeletal system and talking about cartilage also as far as being supportive and part of that system there, and again, protecting bone. And really know that also that hyaline cartilage is the model for the embryo. Really, as the embryo is developing in the skeletal system, it begins as a hyaline cartilage model that is replaced with bone, and then the remaining portions of the hyaline cartilage would be on the ends of the bone and the growth plates. So hyaline cartilage, very unique in its appearance. Okay, so now we're going to be moving on to bone. And there are two types of bone. So let's look at compact and spongy bone. All right, folks, so we'll see here, compact and spongy bone. So as we're looking at compact and spongy bone, there's going to be examples of, let's see here. So this is a good image just to take a look at for a moment here. And just realize that so as we're looking at this image, we can see that there are different types of bone. We have compact bone, which would be this layer right here, would be considered the compact bone, these two layers. Between the compact bone would be this area where we have holes, right? And so this would be the spongy bone. So this area contains trabeculae. These are rods of bone. That's spongy bone. So when we think of a sponge, you think of that there's holes in present, and that's what you think as far as spongy bone is concerned. It's also called trabecular bone, or cancellous bone is another term, right? Trabeculae, these rods of bone. Throughout these rods of bone will be blood vessels, will be marrow, okay? We'll talk about that in a moment, red and yellow marrow. Here you're looking at this compact bone. It's called the osteon, okay? And it's a structure that we're going to look at. Let's take a look here. Let's see. We'll look at compact bone histology first. Okay, all right. So what you're going to see here, folks, is very interesting, and I'm going to show you. Right, I'm going to show you this image first. So as we're looking at this image here, this is compact bone, and this structure right here is called the osteon, and it's concentric layers of bone. Concentric layers of bone. So as you look here, can you see that there's an opening right here, and then there are these multiple layers of bone, these lamellae, these layers of bone, okay? And this space right here is where is going to contain blood vessels, lymphatic vessels. Those structures are going to be nerves would be present within, nervous tissue would be present within that hole, that central canal, okay? Let's see here. Here's another image here. And you're seeing how they've showed you where these concentric lamellae, these layers of bone are present. This is compact bone. Compact bone, now I'm gonna come out of the 400 magnification just to show you a little bit more regarding compact bone. Let's see, let's do this. So as we're looking at this image here, what we're seeing is that this is a long bone, a shaft of a long bone where we've taken, removed images so that we can see that this outer area is called compact bone. The inner area, right, in this medullary canal is called spongy bone. So you see they're both in different locations. Know that also spongy bone can be located at the ends of bone. Spongy bone can be located at the ends of bone, and it's also, you see how it's porous, so that, or not porous, but there's openings. These openings will allow for then the marrow to be present here, as well as here in the medullary cavity. Okay. So looking at this image again, in this space right here, this would contain bone marrow. Okay. And primarily it's going to be yellow marrow. The ends of bone will contain red marrow. Also flat bone, like the skull, will contain red marrow. This red marrow produces blood cells. It synthesizes blood cells. The yellow marrow present within the medullary cavity, this is adipose tissue. Adipose tissue, and when there's a need for more red marrow, this yellow marrow can become red marrow. Okay? If we need more blood cells being produced, being synthesized. Let's show you just another image of. Okay, so here we have compact bone and we have spongy bone. And you'll see in the spongy bone, you'll see that the blood vessels, the vasculature, weaving in and out through these holes. So the yellow marrow is adipose tissue, and it's just present there for in case that we need it as far as nutrition is concerned, as well as just allowing for then filling that space so that it's not just hollow, but that you have the marrow present in there. That's good enough for now, okay? So that would be yellow marrow primarily in the medullary cavity in this tube right here of long bone, the inner portion of the long bone, the red marrow on the ends of the bone for the blood cell production, okay? Now look here as far as this compact bone. So you see that you have these areas. If you were to take a cross section, it looks like almost like a log was cut. That's what we would think of when we think of compact bone, okay? And you'll see that these are, compact bone really consists of these long tubes of bone. Let's look at this image here. And what you're looking at here is, this is that osteon, and it's going the length of, so I'll bring my paper towel roll here, right? So if this represented the shaft of a bone, well, the osteons, these compact bone, they would have these long tubes of bone for the actual shaft of the bone. So compact bone, it's all tightly packed together. And within the compact bone, you'll see here that, look at this image. It's adipose tissue, the yellow marrow. You'll see here that there are lacunae, there are spaces, and in the spaces are osteocytes, bone cells, okay? So they're not bone cells here because that would be the layer of bone, the compact bone, but here we would have these spaces, and these spaces contain the osteocytes, okay? So do you see how it's different than epithelial tissue? Epithelial tissue is tightly packed cells. Here we have cells within a matrix. That's connective tissue. So that's the compact bone. Now the spongy bone, here. Okay, so what we're looking at here, I'm going to take a view that's a little bit like at a 100 magnification. And what you're seeing here is that these pink, see the pink lines? These are the trabeculae, the rods of bone, okay? The rods of bone that's the spongy bone. Anything in between, in those spaces there, would be marrow and vasculature, okay? Marrow and vasculature present in these spaces, but the pink, thick pink, that's the spongy bone. So let's take a look at, so here we go right here. So here what we're looking at is, we have here then the spongy bone, and then in here would be the marrow space. See that there? Marrow, okay? Look at, here we go. Here's another one. We're looking at, here's the spongy bone, the trabeculae, it's the rods of bone, okay? and then we have the marrow. So that would be compact and spongy bone. So now the last connective tissue that we're going to be looking at would be blood. Okay. So when we're looking at blood, folks, I want you to notice that this is what kind of looked like in the lab there. What you'd see is that primarily it's erythrocytes, red blood cells. Okay. There are then interspersed, see the P here? That's a platelet right there. See, it's a little tiny little cell present. And then you have these different colored pink, purple stained cells. These are specific types of white blood cells, also known as leukocytes. So this is a blood smear. So it's very unique in its presentation and what it looks like. Okay. Here's another image. Again, here's 2 white blood cells, and then we have the erythrocytes. And I think that looks like a little platelet right there. Okay. Let's see. Here we go. Leukocytes, platelets, and the rest are erythrocytes, red blood cells. So white blood cells, the majority are red blood cells. That's a blood smear. Blood smear is pretty easy to identify and to note that that's definitely looks like red blood cells. It looks like, you know, that's blood. Pretty simple to identify as far as tissues are concerned. Okay. So we've gone over connective tissue. Now I'm going to show you the different types of muscle. I'm going to stop sharing for a moment, just see question-wise and input here. Yeah, and I mentioned that before as far as that the yellow marrow is fat. Okay, right, good. Okay, excellent. And so that's all connective tissue, that's correct. Yep. Okay, let's go back to sharing my screen with you all. And now what we're going to be looking at would be the types of muscle. So let's look at skeletal muscle first. Okay, so skeletal muscle, very unique in its appearance, as many of these cells are, and it can look a little similar to the connective tissue as far as the dense regular, but you'll see here that there's really, you know, it's like they're layered on top of each other, and there are these striations. See how these lines are present? Okay, we would say that skeletal muscle is striated. Okay, take a look at this image here. These are skeletal muscle fibers, and you see how they are striated. Okay, striations. So cardiac muscle and skeletal muscle both have these striations present. Okay, and here you're looking at these striations. Okay, skeletal muscle. Now, cardiac muscle, really unique part of the anatomy of looking at cardiac muscle would be notating the, let me just show you what looking at. You're going to see these intercalated discs, okay? So when you're looking at skeletal muscle, cardiac muscle, and I'm going to put cardiac. Okay. And so this is something, this would be approximate to what we, oh, Okay, what question do you have? Who has a question for me, and I'll answer. Does anybody have a question right now? And
I
I
do.
stop sharing.
Well, it was going back to the eustachian tube. Can
Yes.
you just like spell it out later?
Sure. Yeah. You can. It's EUSTACHIAN.
Uh-huh. Oh, okay.
Eustachian. Okay.
Okay, thank you.
You're welcome. So you'll see here with this image that you'll see that it has those striations, and you'll see that it has these dark lines present also. Well, these dark lines are unique to cardiac muscle. Okay, cardiac muscle. Let's look at this one right here. And here you can see the dark line. So there are lines present, there are striations, there are these dark lines present. Let me go back to this one again. And then you can also see there can be, depending upon the slide, this one shows it, that they're kind of branching and such. A little bit different. So cardiac muscle, unique as far as having those intercalated discs, and there's branching present within the examples of the tissue. Yeah, here's another one, right? So it's different. It looks similar to, but it's different than the skeletal muscle. And again, it's got these darker lines, that are interspersed throughout that allow for cardiac muscle to different areas. So the atria, the upper chambers of the heart, and the lower chambers of the heart, the ventricles, contract as one. Okay, and so those intercalated discs are part of that whole connectivity of the cells of that tissue. So cardiac and skeletal muscle, they're striated. Skeletal muscle is voluntary, cardiac muscle involuntary. Lastly, we're looking at smooth muscle. And for smooth muscle, let me show you this image first. Now, smooth muscle, we say that they have these spindle-shaped cells, okay? So take a look at this image right here. So this is really what these smooth muscle cells look like, okay? Now, when you look at them as far as in the lab and on images, what you're seeing here, folks, is that, and how I express to my students is that you're looking at, imagine it's a cell that has the nucleus that's been stretched. So the nucleus is elongated, okay? The nucleus is elongated. And so do you see that here? So are you really seeing the differentiation between, you can see a little bit of like these, it's hard to notice them, but you can see this right here. But primarily, if you just notice that, hey, those nuclei look like they've been stretched, that's smooth muscle. Okay, that's one of the a major indicator for smooth muscle. So you can see that here, kind of, that's not as good. Here, see those? Let's look right. So you see how this, on this image, it looks like those nuclei have been stretched a bit. Smooth muscle. That's when I'm testing my students, that's the kind of presentation that I show, as far as so that you're knowing that, hey, that looks like different from what I've seen with the other samples, and that looks like those nuclei have been stretched. Smooth muscle. Okay. Right. So we've gone over the 3 different types of muscle, and smooth muscle would be involuntary, right? I have no conscious control over smooth muscle. And what'll help you is that when you're doing this, when you're looking at these different images, that you go back and forth and review images to see that, hey, I've got these will look a certain way, and it's very distinct in their look in comparison to. So when you're looking at, I would focus on looking at all the cartilages together, the bone together, the muscle together, right? So this way you're looking at groupings of tissue, the epithelial tissue. Look at the simple ones, look at the stratified. I mean, but That's how you have to like break it down into smaller groupings of the information. The last slide that I'm going to share with you would be a neuron smear. Okay, so when we look at this neuron smear, and I'm first going to just show you, yeah, so this is when you're scanning and looking at this, folks, is a neuron. All these other little cells here, supportive. They are glial cells. They are support cells. But this is a neuron, okay? Has multipolar neuron, so it has multiple dendrites and an axon, okay? Very important that you are able to recognize the neuron smear. So here we have, this is pretty cool. So you're looking at, these are dendrites receiving information to this neuron cell body. And then the axon hillock is where the action potential will then reach out to the axon and go to a muscle or a gland. Let's look at another image here. There we go. So here you're seeing, and it's okay also when you're doing your Google search to look for images that have labels, identifying structures and such. Can be very helpful, okay? So here you're having these support cells surrounding this multipolar neuron. These are dendrites receiving information. They're called graded potentials to this nucleus. And the axon is sending out an action potential to another neuron, another nerve cell, or a muscle or a gland. These are all nuclei. All nuclei of neurons, okay? So you're seeing the inner portion. So better, here we go. So here's the nuclei present within this structure right here, which is the neuron cell body. So neuron cell body here, 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 right? Containing the, see the cell body, containing the nucleus and other structures that would be present in a eukaryotic cell, right? Lastly, I want to show you a slide that we don't have on in our lab, reticular. Reticular tissue is the type of tissue that is located in the lymphatic system, okay? Reticular connective tissue. And so what's going on? It kind of looks like grapevines with grapes attached to them, okay? Reticular connective tissue, again, we don't have it in the lab that as far as testing is concerned, but I want to show it to you that realizing that lymphatic tissue comprised of reticular connective tissue. Okay. All right, now let's see here. Let's stop sharing for a moment. Question. Okay. All right, so are there any questions regarding what I've gone over today as far as the histology and the tissues are concerned? Again, I've posted chapter 5 for you all, so this way you can review that and go over it. And I'm going to go into chapter 3 next, chapter 3 and 4 PowerPoint. Again, we didn't have the opportunity to have the class. I did share the recorded class I had for your sister class, but we didn't all get a chance to have class on Monday. So I'd like to go over some information for you from chapters 3 and 4. Any questions at all? And I can only go 20 minutes till 12:20, but we'll review some information. So let's go for it. Okay, so let me minimize this. Let's go to here. All right. So what I'd like to do is I would like to cover the latter part of the information from the. I'll go into the slideshow. Let's start right here. I'm going to start right here and try to go to the end of the PowerPoint. Again, I've covered it in the PowerPoint lecture that I recorded for you all. I want to hit the end of it, okay, by showing you this because we're not meeting for class the rest of this week. So I'd like to cover this before we leave for today. So I'm going to be going kind of quick because again, I have another class and you have other classes to go to. So we'll go for a few minutes there and review information. So active and passive transport are types of ways that the body, particularly the cells, can allow for information materials to go in and out of the cell. When we look at the cell, in particular the plasma membrane, this phospholipid bilayer, it's a fat-containing membrane, right, that covers the cell. We have the extracellular fluid and we have the intracellular fluid. This intracellular fluid contains the cytoplasm, which we would say would contain the cytosol, the liquid portion of the cell, as well as organelles, and also the nucleus with its genetic material. Now, material that can move from a high concentration, you see how this is a larger letters than this one right here? So high concentration to low concentration via this protein channel, leak channel, it's moving from high concentration to low concentration. No energy required. ATP is required for when we need to go against the concentration gradient. So think of that high concentration to low concentration. There's diffusion. It's moving from high to low. No energy is required. But when we go from low to high, that's a problem. That you need ATP, you need energy in order to move from low concentration to high concentration. So know that sodium and potassium ions are located on both sides of the plasma membrane, in the extracellular space outside of the cell and inside of the cell. It's just that the concentrations are different between the two of them. Potassium ions are higher in the concentration inside, sodium ions are higher concentration outside. So going against that concentration gradient requires ATP, requires energy. You'll see here, as far as these brackets represent concentration. So movement from high to low, that's important for you to know. So here we have high concentration, low concentration, right? And know that this is a selectively permeable structure, the plasma membrane, okay? So movement from high to low will create over time equilibrium. Now, this is equilibrium where it's equal concentration on both sides, but high concentration, low concentration, movement from high to low. Okay, very good, no problem. Thank you. Osmosis is movement of the solvent, not the solute. So solute, you can think of these blue particles here. They could be glucose, they could be salt, right? They could be sodium ions, they can be whatever it may be. But in the case of potassium ions are higher concentration outside of the cell, then let's think of them as sodium ions. Moving from high to low, that's through the constant. Now really, and because there's no, you're not seeing any type of protein channel, think of it more as like another substance that would be present that would be able to diffuse easily across the plasma membrane. And just for example, diffusion from high to low, it's considered they lose concentration when they move into the cell. Now, it just means that when I'm thinking of the extracellular space, high concentration. When I'm thinking of the intracellular area, low concentration. This is equal concentration. You see that? And we're looking at the solutes. We're not looking at the white would be considered the solvent. The solvent many times is water. Okay, so diffusion goes from high concentration to low concentration, whatever it may be. We looked at sodium and potassium ions. You can also see here as far as oxygen, moving from high to low, diffusion. Osmosis is the diffusion of the solvent, the water, from high to low concentration. So look at this here, folks. Here we have this semi-permeable barrier, and we have high concentration of water, low concentration of water, the pink, not the little dots, the pink. That would be the solvent, the water. So moving from high concentration to low concentration, you have equilibrium now. Although the concentration is equal, not the amount. It's the concentration of the pink, of the solvent. That's diffusion of water, osmosis. Okay, yeah, these are concepts, folks, that you have to review and go over, and it takes a little bit to understand it, okay? But it's okay, you'll get it. Now, tonicity. Tonicity deals with what's taking place with the solvent. Now, in the case of the blood and red blood cells, in a hypertonic solution, and this is why you cannot drink salt water. So if you were stuck on a desert island or out in the ocean and you're drinking salt water, it will kill you. It'll be a painful death, actually. You don't want to do that, right? A little bit of salt water, not a big deal. If you're gargling with salt water in order to help with like a sore throat, okay. But drinking salt water, never a good thing. It'll create what's called a hypertonic solution in your blood, and the blood will have too much salt present within, which will in turn lead to the red blood cells crenating, shrinking, and dying. Hypotonic solution. So if you drink gallons of water in a row, you just force yourself to drink a lot of water all at once, you could die as a result of that also, creating a hypotonic solution. So the red blood cells will swell, they'll swell and they'll burst, lysis. That's what we're talking about there. Isotonic, if you use saline solution for your eyes, I did contact lenses for years, use saline. This is a slightly salty solution, right? Very low salt content, but it's salty a bit. This is the case of what's taking place within the blood, in that it's just got this type of solution that's called isotonic, and it will not affect what takes place with the red blood cells. Let me show you an image. So here we have that very salty solution. So the plasma of the blood, because of drinking too much salt water, will lead to this hypertonic solution in the gray here, and the cells will shrink. All the water from the cells, you see it here, will exit into that hypertonic solution, and the cells will die. Or in the case of where there's a very low salt concentration, hydrate patients, yeah, because then that's involved in aiding in the increase in the actual plasma. So if someone, if they're low in the amount of, so that if it was a situation where they were bleeding and a catastrophic accident and such, and they lost blood, so a quick way in order to actually help to increase the blood volume would be to do that, okay? So hypotonic solution, this can lead to the cells bursting, right? Because then water would enter in. Isotonic, it's creating an environment where it's slightly salty, right? And there's water will move in and out of the cell, but it will not damage the red blood cells. If we damage the red blood cells, then you're gonna have issues with the actual transferring of oxygen to the tissues of the body and removal of CO2. Very bad. Tonicity. Active transport here, again, requirement of ATP. Passive transport just moves from high concentration to low concentration. You'll see here, as far as there are also methods using carrier proteins. And a carrier protein will in turn either move one substance in one direction, or move two substances in the same direction, symporting, or antiporting, moving one substance in one direction and substance in the other direction, opposite. This is the case of the sodium-potassium pump. It's an antiporter. See how sodium is going from intracellular to extracellular. It's going against its concentration gradient, so we need ATP. And potassium is going from extracellular to intracellular against its concentration gradient, requiring ATP. So as sodium moves in, potassium moves out. Or, I'm sorry, as sodium moves out, potassium moves in. Correcting myself there, sorry. Okay, so here we have, and this is explaining to you with the antiporting, 3 sodium are moving out, 2 potassium are moving in. Again, they're moving in different directions, and so that's called an antiport. Now, the channel proteins, these are proteins present within the plasma membrane, that fluid mosaic model, right? That we have leak channels and we have gated channels. Leak channels always open. There's material moving. Sodium is moving from outside in, down its concentration gradient from high concentration to low concentration. We saw that already. Gated channels will allow for movement of material, but you have to have a stimuli in order to open. You need a key in order to unlock the gate. So in the case of ligand gated, we need a molecule. So like in the case of at the neuromuscular junction where we have nerve and muscle interact in order to cause muscle contraction, we need a neurotransmitter called acetylcholine. In the case of voltage gated, we need a change in the charge of the plasma membrane in order to, so there needs to be a little bit of a change in the charge, and that'll be the ability to, the key to open that voltage gated channel. So here we have leak channel, we have a gated channel, another gated, this is a leak channel, okay? So these are always open. The gated channel needs a key in order to open. Oops. All right. So here you're having these ligand gated channels. We need a specific, and they're specific, folks, a specific molecule, chemical that will interact. So here we have this acetylcholine that will act as the key to open the gate. Voltage gated, there needs to be a change in the voltage. And so know that a little bit outside of the cell in the extracellular region, it's positively charged. A little bit inside of the cell, right in the very inner portion of the intracellular region of the plasma membrane, it'll be negatively charged. When this charge changes, that's going to cause then allow for a voltage gated channel to open. I know it sounds kind of weird and such, but just know that you'll see here as far as it deals with millivolts, right? And so you see here negative 70 millivolts, negative 50 millivolts. There's been a change in the voltage, and as a result, it's opened up the channel. So you see here, this was the gate. Here's the gate, and now it opens as a result of a change in the charge. And mechanically gated, this can be as a result of, so you see here, pressure that's being applied. Sorry, pressure is being applied. So there's actually some type of mechanical movement that's taking place that will also aid in opening up this gated channel. The G protein complex is important in that if I don't have the ability to enter into the cell, passing through the plasma membrane, and there's no gate present to do that, we can still cause change to take place within side of the cell in the intracellular region. So read this here, interaction with the receptor in the extracellular region, so outside of the cell, can cause changes in the intracellular environment inside of the cell. So water-soluble proteins cannot pass through the plasma membrane, right? It's impermeable to that. So what happens then is that these hormones will interact with a specific receptor for that specific hormone outside of the cell. It's still attached to the plasma membrane, but it's on the outside, which will cause change to take place on the inside. Okay, so you're seeing here, here's that signal molecule, and it's interacting with this receptor on the outside of the cell to cause change to take place inside of the cell. Just giving you some examples of. Let's see here. Okay, good. All right. I'm sorry that I've gone through this quickly, but I will not be with you all next week, so I wanted to at least finish this for you. We're not meeting in class. I will share the video for you, the link to the video for the class that I teach tomorrow in the afternoon as the part 2 also. So this way you'll have this, what I'm talking to you today, as well as what I've done for the other A&P1 class, okay? So you'll see here that substances entering into the cell would be entering via endocytosis. They're entering. Substances leaving the cell, they would be exiting exocytosis. Yes, Tara, she should be coming back. Dr. Massimo will be back next week, as far as I know. I don't think there should be any issue. We'll let you know if there's any type of change. You will need to know these different types of terms describing endocytosis, material entering the cell. Okay? Pinocytosis for liquid material, phagocytosis for material that's not a liquid, okay? And receptor mediated for specific receptors that will allow for then information to pass into the cell, information, molecules, whatever the material may be that will pass into the cell. exocytosis, it's leaving. So enzymes, mucus, breast milk, these are all leaving the cells of the different respective areas of the body, in the case of the breast milk, breast tissue, and then exiting the body via exocytosis, exiting the cells that are producing the secretion. Okay?